Ah. Okay, so last time we had a bit of an intro into Streptococci. We talked about hemolysis and we talked about what Lancefield groupings are. Lancefield groupings basically just a serological classification of bacteria based on the bacterial antigens they have on them. In this video we're going to go over the group A and the group B Lancefield groups for Streptococcus. So group A strep, for example strep pyogenes, can be identified by 1 to 2 millimeter white colonies producing a beta hemolysis on a blood agar. So that basically just means they're producing these hemolytic enzymes breaking down the red blood cells. They are sensitive to the antibiotic bacitracin, and they are pathogenic strains that tend to be capsulated. Strep engage in pathogenesis, so the evasion of phagocytes, through use of either protein M, which interferes with complements responsible for opsonization of the strep pathogens, or through the use of a hyaluronic acid capsule. Sorry, one moment. Through the use of a hyaluronic acid capsule which camouflages the strep bacteria from white blood cells. Strep can also produce two kinds of membrane-bound proteins called streptolysins, which lyse red blood cells and white blood cells, so breaks down red blood cells and white blood cells, interfering with erythrocyte oxygen carrying and blood clotting, and destroying any leukocytes that try to phagocytize the strep bacteria with these streptolysins. And then once these phagocytes have engulfed these streptolysin-bearing streptococci are lysed and they're destroyed, the strep will have proliferated and they'll be able to be released back into the blood and then infect more cells, and then it's where you get your problems like bacteremia. Oh, is that where we were? Sorry about that. So strep produce two kinds of streptokinase enzymes to break down blood clots for faster spread. They produce 4 kinds of deoxyribonucleases to break down and depolymerize DNA for faster spread. And they produce a C5a peptidase to break down the C5a complement responsible for chemotaxis. And if you don't remember, chemotaxis is just the recruitment of immune cells to the site of infection. And they also produce hyaluronidase to break down the hyaluronic acid for faster spread through tissues, same as Staphylococci. So group A strep, so for example strep pyogenes shown here, secrete 3 distinct pyrogenic toxins that stimulate macrophages and helper T cells to secrete cytokines that stimulate fever and shock. Only bacteria that have been infected by a temperate bacteriophage possess the genes responsible for the production of these toxins, called lysogenized bacteria. Group A strep tend to infect the pharynx or the skin, causing temporary abscesses that exist until the immune system begins producing antibodies for these M proteins and streptolysins that we mentioned in the previous slide. So this is what one of these abscesses look on the top right here, full of these streptococci bacteria. The infection can then reach the deeper tissues as the bacteria proliferate in the abscesses. The bacteria can spread through a number of means, such as respiratory droplets, and they are treated with antibiotics. Group A strep can cause multiple diseases depending on where these Streptococci bacteria spread to. So a strep infection of the throat causes throat infection, which is officially called pharyngitis. A wide Streptococci infection of the skin can cause scarlet fever. You can get streptococcal toxic shock syndrome, which I won't go into too much detail in. If you want to learn what toxic shock syndrome is, have a look at the video, the second video on staphylococci, where we talk about the toxins that they produce, because we talked about how staphylococci produce toxic shock syndrome toxin. And then necrotizing fasciitis here, which is where the streptococci infection has caused inflammation and reduced blood flow to tissues, starving them of oxygen and killing those tissues, and then these tissues become necrotic, and you get a lot of saprotrophic bacteria. They're going to be cultured in these dead tissues, and they're going to spread, and then the infection is going to get worse, and it can be very dangerous. This can't be treated with antibiotics because antibiotics require active blood flow to reach the bacteria. So if it gets to the point of a necrotizing fasciitis, the only option is surgical removal of the necrotic tissue, treatment with antibiotics to prevent spread of the bacteria, and you need to hope that any antibiotic resistant bacteria or bacteria that we can't treat through antibiotics haven't made their way through and spread to more important tissues, because we can see the necrotic fasciitis of the lower leg here. It could be any tissue, it could be your abdominal region, and obviously if you get necrotic tissue in an area surrounded with important organs, that's going to be a lot more of a problem. So group A strep is diagnosed in the lab with a blood agar for hemolysis or observed under the microscope. So like we mentioned before, if we're looking for strep, we're going to be expecting a beta, if we're looking for group A strep, we're looking for a beta hemolysis. Treatment tends to be penicillin, just a regular antibiotic, or alternatively, if you're allergic to penicillin, you may be prescribed erythromycin or cephalexin. So that's it for the group A strep. We're running out of time, so I'll make the group A strep its own video, then I'll make the group B strep its own video, and then we're done on the Streptococcus. Thank you.